Hello everyone. Viruses are interesting living beings. They are small, cryptic and eerie. These puzzling microbes are a challenge for scientists and philosophers alike. They seem to be parasitic in nature but are not parasites. They can neither create nor utilize energy on their own. They are almost lifeless on their own but come to life when they enter other cells. Philosophically thinking, it's very difficult to determine the purpose of their lives. They possess the same atoms that are abundant in living organisms like carbon, oxygen, hydrogen and nitrogen. Since these atoms are versatile in their chemical interactions, unlike metals or minerals, viruses can interact easily with living cells. They have genetic materials which make them to qualify to be part of animate objects. Their ability to replicate into millions of cell copies make them indisputable as living organisms. On a lighter note, it's still amusing to me when I think of their purpose in life. They can't eat, think, sing or dance, but how easily they can make us dance to their tunes. As we know, there are viruses which infect humans and animals, and there are others which infect plants, and some of which find their bigger brothers, bacteria, as their hosts. The first virus, which in Latin means poison, was identified on tobacco plants, called mosaic virus. In fact, viruses infecting bacteria are called bacteriophages, and many bacteriophages were in use to treat infections in East European countries. So that makes viruses also as helpful creatures to humans. Well, that's a new perspective towards them. As we move, we will also learn that viruses might have significantly contributed to our evolution by infecting different individuals and distributing their DNA within a species or from one species to other. For example, retroviruses incorporate their DNA into the host cell and later take a piece of the host DNA while multiplying. This picture is not to the scale. However, this picture is basically to understand that viruses really are very, very small, tiny living beings compared to a bacteria. And bacteria themselves are very small compared to an animal cell. It's always easy to remember things in relative terms, and hence I will explain a bit about bacteria and their structure and relate them to viruses. Bacteria have a thick cell wall with genetic material clumped together inside it in a part called nucleoid. They do not have nucleus. Bacteria have pylosis. These small hairy structures which help them in floating while the long tail generates the movement. Bacteria can generate their own energy, they can generate their own proteins, and they can multiply by a method called fission. They break into multiple pieces with equal amount of organelles of the cell in each piece. But viruses can do none of these. They need to penetrate into the cell and use all the materials available inside the cell to multiply. Interestingly, sometimes they cannot penetrate the cell wall of bacteria, and hence they just inject their DNA or RNA into the bacteria through a small hole in the cell wall, and genetic material starts becoming active. Viruses are very, very small and can be present with just a nucleocapsid when they are called naked viruses, or they can have an additional membrane covering the nucleocapsid as an envelope. The coronavirus, which is making news now, is a virus with an envelope. So they are not naked. The capsid around the genetic material is very artistic with beautiful patterns, colors and shapes. Wonder whether they can appreciate each other. Just kidding. You can think of a copper wire tightly held by a plastic material and presented as a small coiled piece as the structure of naked viruses. Since the nucleotides which make the genetic material are tightly held by the capsid, the unit is called nucleocapsid. Some viruses have additional features like crowns in the coronavirus or thorn-like protrusions in hepatitis A viruses. They sometimes use these additional proteins to bind to receptor on the animal cell and gain entry into the cell. Virus DNA or RNA is linear, and center of the classification of viruses is based on the genetic material of viruses. If we recall genetics, DNA codes for proteins and other biochemicals which are required for life. The part of DNA which codes for the specific biochemical required for the cell gets created by transcription, which is called messenger RNA, shortly called mRNA. It is mRNA which is, with help of tRNA and ribosomes and few other apparatus of the cells, starts journey of life of the virus. mRNA is at the center of classification of viruses. Let's try to remember this simple fact for the understanding of classification of viruses. The viruses can have their genetic material as DNA or RNA. The RNA present in the viruses could be in the form of sense, which means it can directly create an mRNA which can create proteins. If it is the opposite, it is called antisense, which basically means the opposite strand needs to be generated, which in turn can code for the proteins. I will give you a simple technique to remember 7 groups of viruses and which group has maximum members who infect human beings. Count 1 to 7 starting with major parts of our body. While head is 1, 2 upper limbs are 2 and 3. Chest is 4, abdomen is 5 and 2 lower limbs are 6 and 7. This has absolutely nothing to do with the viruses and the parts infected. While the numbers representing head, chest and abdomen make the most of viruses and rest have very few. So group 1, 4 and 5 are the largest groups. The classification of viruses is based on DNA, RNA and retro. There are 2 DNA virus groups, 3 RNA virus groups and 2 retrovirus groups. Let's look at the groups now. DNA double strand, DNA single strand, RNA double strand, RNA single strand sense, RNA single strand antisense, RNA retroviruses, DNA retroviruses. Now that looks pretty simple. Read it twice and you will remember it. As I already said, group 1, which is double strand DNA viruses, is a big group. Many viruses in this group infect skin, for example herpes, smallpox, human papilloma virus or HPV, which is known to cause cervical cancer, belong to this category. 2 other members are CMV or cytomegalovirus, which is a cousin of herpes virus, and Epstein-Barr virus, which you can remember for kissing disease, and a post-infective demyelinating disorder called Guillain-Barré syndrome. So skin infections plus CMV and EBV are the common infecting viruses of this group. Single-strand DNA viruses rarely infect humans, so group 2 can be neglected. Another easier way to remember could be that DNA is double-strand and double-strand DNA viruses are common, while single-strand DNA viruses are not common. Similarly, RNA is single-strand and hence double-strand RNA viruses are not common, while single-strand RNA viruses are. So in group 3, that is double-strand RNA viruses, are not many, but rotavirus is the major pathogen of human infections due to viruses. Rotaviruses are notorious in causing diarrhea in children. A vaccine is available against this virus. Group 4 contains single-strand RNA viruses which are positive or the RNA strand is sense and can directly code for proteins. There are several viruses in this group including coronaviruses. Hepatitis A, C and E belong to this category. Please note hepatitis A and B do not belong to the same group. Dengue and chikungunya, rhinovirus, rubella virus and yellow fever virus also belong to this category. So is poliovirus. Group 5 is a big group again, single-strand antisense RNA viruses. Basically means that this RNA strand needs to create a complementary strand which can work like mRNA and produce proteins. 3 common and 2 deadly diseases belong to this group. Rabies and Ebola are the deadly ones. Influenza, measles and mumps are the common but less virulent diseases. Group 6 has single-strand RNA retroviruses, of which notorious is HIV virus. Group 7 belongs to double-strand DNA retroviruses, of which hepatitis B is very important. To sum it up, Group 1, 4 and 5 are big groups. Group 1 basically are skin with CMV and EBV. Group 2 is non-existing. Group 3 is occupied by rotavirus. Group 4 is a master group with corona, polio, dengue, chikungunya, rubella, hepatitis A, C, E and yellow fever virus. Group 5 has rabies and Ebola along with influenza, measles and mumps. Group 6 is HIV and Group 7 is hepatitis B. Now let's understand the mechanism of entry of virus into human cells as well as how they cleverly utilize the cell substances for making their own copies. Viruses have different mechanisms to enter the human body. Some of them are vector-borne like dengue, some of them are transmitted through fecal-oral route like hepatitis A, and if you have noticed, hepatitis B is totally from a different group of viruses and hence gets transmitted only through blood. Whatever is the mode of transmission, they should enter the body cells to come to life. There are 2 primary modes through which a virus enters the body cell. Many viruses are, as shown in this picture, have envelope protein which is part of the animal cell protein. Hence, they easily fuse into the animal cell and enter the animal cell. Some viruses use receptors present on the cell and bind to them. The moment the receptors are bound, the cell opens the gates on their cell wall and lets the virus in. If you keenly observe, those additional marks in blue and red in this picture denote receptor and the receptor binding protein on the virus for the virus entry. SARS-CoV-2 virus, which is responsible for COVID-19 disease, enters the cell by binding to ACE2 or ACE2 receptors. As you can see in this picture, once the viral genetic material is inside the cell, it immediately starts using the transcription enzymes of the cell and starts making several of copies. As it makes copies, it also generates toxic substances which lyse the cell and ultimately cell dies, releasing several baby viruses or virions. Again, these are lifeless virions which are ready to infect the next cell in the same body or another body. There are few viruses which do not go on to lytic phase as soon as they enter the cell. They lie dormant inside the cell and go on to replicate at an appropriate time. Humans with such viruses are called carriers of the virus. Though they do not have the disease, they can still transmit the disease. You know the carrier stage of hepatitis B. This is the slide to show you some special aspects of retroviruses. As you know, this picture demonstrates HIV virus and its cycle. You can see GP120 protein here, which binds to CD4 receptor on the cell and fuses with the cell. These viruses bring genetic material along with few special enzymes which help them to start replication once inside the cell. For example, HIV has an enzyme called reverse transcriptase which can convert viral RNA to DNA as soon as the virus enters the cell. It also has another enzyme called integrase which integrates the viral DNA with the human cell DNA. And as you know, the DNA is master of the cell, and once the viral genes get inserted, they take control of the cell. They can also integrate these genetic materials to other cells. There ends the first part of this lecture series on viruses. Please write to AtrimedX at atrimed.com if you have any queries. We will answer the queries and post them as soon as possible. Thank you.